either IPTG or tetracycline, as well as chemical temperature toggles that are shown on the right that could be induced with either IPTG or heat shock. Shown on the right is that we actually could get bistability, showing 4 different bistable systems that can start in the off. You'll whack it with or induce it with IPTG, flips it into the on state. You can remove the inducer, it stays there. Then you can give heat shock and it'll go from on state to off state. Each of these states was highly stable, producing then a highly functioning circuit. Shown on the bottom right is that the circuit actually behaved with a very sharp switching threshold, which opened up the possibility that it could be used for biosensing. Going back now 20 years ago when we published this, it's hard to believe it's that long ago, there was a lot of interest in thinking about how you could use this programmable memory, this now programmable capability that we and others showed, to create living computers. And I think that was a wrong notion and remains wrong for many reasons. And if we have time for discussion at the end, we can enter into an exchange. And instead, I think what this work and others set up was this idea that we could create programmable cells. Cells that you could upload these synthetic circuits, which would endow them with the ability to detect an input of interest from their environment, make a decision on that input, and produce an output that could impact the environment. And these programmable cells have then opened up the possibility of living technology that's really beginning to dramatically impact our world and all different swaths of the economy, including agriculture. So there are groups now using the toggle switch, for example, to create drought-resistant crops where you can keep the drought gene, which might impact the fitness of your crops, in the off state, but you can then flip it into the on state with the transiently delivery and inducer by dusting your field. In the environment, there's a lot of interest now in programming bacteria to sequester carbon from the atmosphere. Further interest is, can you program bacteria, for example, to endow corals?
Sorry, Jim.
Yes.
Sorry, Jim, to interrupt you. It is outside from the presentation mode now. Please, will you go to the presentation mode again?
So I did not leave the presentation mode, but let me see.
It was previously in the presentation mode, but someone has created a problem. So can you start from the presentation mode again?
Let me see.
Yeah.
That work.
So click on the slide. Click on the slide, please.
Okay.
Yeah. Go to the full mode.
So.
Yeah.
How's that?
Thank you. It's okay, fine.
Okay. And so please jump in at any time that there is a technical problem. I did not recognize that had happened. And do not hesitate. Don't be too polite, because I want to make sure everybody can see what they need to see. So are we fine right now? Okay. So also from the energy standpoint, the idea was, could you program bacteria to convert sunlight or various foodstuffs, glucose, in order to make fuels? And it became a very big interest in the field going back to the mid-2000s. And hundreds of millions were poured into the field. But it turned out that we could at best make $1 worth of fuel for $8, which was not a great business model. And that focus of the field imploded on itself. What has emerged in the last decade, and we've played a role, is now the idea of using living technologies for biomedical applications, including the current pandemic. We were challenged by the Gates Foundation a few years ago. Could we engineer bacteria to detect and treat cholera? As many on the call know, cholera is a nasty gut pathogen that has troubled humans for centuries. It essentially is transmitted through contaminated or dirty water. And it's a very clever pathogen that will produce a toxin that will trick our cells into dumping their fluid into the extracellular space, and thus, if left untreated, can lead to dehydration and actually lead to death in the worst cases. We took on this challenge and considered to see, could we engineer Lactococcus lactis, a generally regarded as safe probiotic, to eavesdrop initially on cholera and to then produce antimicrobial peptides that could prevent cholera infections and or treat cholera infections. Briefly, we were able to actually create a living diagnostic by co-opting the quorum sensing system of cholera. So cholera involves two different quorum sensing molecules. We looked at CAI1, which is specific to cholera, took the receptor for that, and created a hybrid receptor that could specifically respond to CAI1 in L. lactis and produce a transcriptional circuit that in the presence of CAI1 and proper detection would turn on and produce an enzyme that would turn stool a different color. Shown in the upper right of this plot is we found that we could have a beautiful dose response curve for this system and that it was highly specific to CAI1 from cholera. Shown below, we actually demonstrated you could do this in an animal model. So in mouse, you could, with high specificity, report out that the animal was infected. We were working on various antimicrobial peptides that could be effective then as a therapeutic, but discovered that L. lactis on its own, unmodified, could actually serve to inhibit cholera infections and treat cholera infections. And it would function by producing lactic acid that would shift the pH of the environment. And as many in this webinar know, cholera really despises a low pH environment, so it does not like an acidic environment. To get a cholera infection, you have to actually drink a lot of cholera to get enough of the pathogen through the acidic environment of your stomach. And so we found that you could actually introduce L. lactis on its own as both a prophylactic and a treatment. And captured here with mouse studies, we showed that you with the administration of the L. lactis could prevent and or treat cholera infections. We found, interestingly, when we introduced then the diagnostic and hope to have then both the therapeutic and diagnostic in a single microbe, that the fitness burden of the diagnostic was sufficient that it actually reduced the production of lactic acid below therapeutic levels. And so to get around this, we actually then turned to just a mixed culture. So we could now mix in wild type L. lactis along with the engineered L. lactis serve as a diagnostic in mice to show that in fact we could simultaneously report out by changing stool a different color and serve to either prevent the infection from taking hold and colonizing the host and or treat the infection. The beauty of this platform is that the L. lactis is inexpensive, easily manufactured, scalable, and thus one could also lyophilize the system. So you could either introduce this into foodstuffs like kefir, or you could have it as pills that could be rapidly distributed at the outset or in the midst of a pandemic, in this case, cholera outbreak. And as many know, we are in the midst of COVID-19, but there's actually cholera outbreaks in Yemen and Haiti that are really bedeviling those communities. We were looking to launch a human trial just before the pandemic hit, and unfortunately the pandemic hit, and that's on hold. But of translational note, we actually moved this technology into a company that was a startup and now is actually a publicly traded company called SynLogic. And SynLogic is using synthetic biology to create a whole new class of therapeutics that they call synthetic biotics, using engineered bacteria to address a range of conditions. So they're treating rare metabolic diseases. They have clinical trials underway. They're treating immuno-oncology with clinical trials underway. They're looking to make a vaccine for COVID-19, and I'll later in the talk present our efforts that are related to it and speak to theirs specifically as well. And they have emerging programs around inflammatory bowel disease, specifically to see, can you engineer bacteria to treat Crohn's disease or ulcerative colitis? In each case, as many know, biologics are the frontline treatment, Remicade or Humira, which are really large sledgehammers on our immune system. So they're immunomodulatory biologics that need to either have an infusion pump every 6 to 8 weeks in the case of Remicade, or a sub-Q needle delivery in the case of Humira once a week. Our vision and SynLogic's vision is, can we engineer these bacteria to deliver immunomodulatory molecules at the site of the disease in the digestive tract, the GI tract, eliminating the need for these complex delivery and freeing up. And the idea would be you could take this once a day or twice a day, really quite harmlessly. And these probiotics would not be modifying your microbiome, so they wouldn't colonize, but they would pass through and produce molecules of interest. And we are now exploring, of interest, I'm sure to many on the call, of how you can also use this to spare the gut microbiome from antibiotic treatment. So can you engineer these to produce molecules that would reduce the killing efficacy of the antibiotic against your commensals, but enable the antibiotic still to kill the pathogen? And I can report, yet to be published, but beautiful work that showed you can actually do that. Shifting now toward diagnostics. We got very excited a few years ago about, could you harness the power and diversity of biology without using living cells? So with my postdoc Keith Pardee, we turned to cell-free extracts. Cell-free extracts have been used in molecular biology for over 6 decades. And it's this idea that you can open up a living cell and take the machinery of that living cell out of the cell and play with it in a petri dish and test tube. These cell-free extracts consist of DNA, RNA, ribosomes, and other molecular machines, as well as other biomolecules such as ATP and amino acids. Keith Pardee had the brilliant insight, kind of captured on the right, left side of this plot, was what would happen if you took a cell-free extract, coupled it with a synthetic gene circuit of like a toggle switch, and spotted these on paper and freeze-dried them. Could you maintain their functionality? And what he found, which was stunning, is that after the freeze-drying, if you rehydrated them sometime later, a few hours, a day, a year, 2 years later, he found that they maintained their activity. That is, they could still function from a transcriptional and translational standpoint, captured on the right here, meaning that after freeze-drying these cell-free extracts and synthetic gene circuits, after they were rehydrated with water or maybe a patient sample, they functioned as if they were inside a living cell, which was remarkable and opened up for us a number of applications that will now be the focus of probably the next 20 minutes of the talk. Our prime interest was, could you use these to create low-cost diagnostics? So we developed a number of RNA switches from a synthetic biology standpoint that enabled us to detect arbitrary or sequences of interest that might be related to an RNA genome of a virus or in an mRNA produced by a bacterial pathogen, or maybe in an RNA element made by a human. We were able to show that these had high sensitivity and high specificity, and that they could produce an enzyme that could change the color of the substrate, for example, from yellow to purple, opening up amazing possibilities for low cost, easy to store, easy to distribute diagnostics. And what I want to emphasize is that this freeze drying then enables others to use biological material without the need for refrigeration, without the need for cold chain. So that you can freeze dry these things onto paper, stick them in your pocket, drive to another locale, stick them in your pocket, get on a plane, fly around the world, stick these just in a regular envelope without need for thermal protection, take them out and use them very inexpensively. And so we got very excited about how this could really change global health in both developed countries and developing countries. And of note, each of these sensors costs just about 2 cents. Much of my lab is focused on antibiotics, and we're going to cover that towards the end, and it's relevant to the current pandemic. And we initially focused on antibiotic resistance. It was our prime focus, and our idea was, could you use these paper-based diagnostics for individuals presenting at a doctor's office or an emergency room in a hospital to identify, is it a bacterial infection, and if so, is it resistant? And we showed you could do this and get an output in under an hour, which is well within the magic window established by infectious disease doctors. This was back in 2014, and captioned on this slide is just before we published this work, and there was a lot of interest from an editor itself. Keith Pardee and Alex Green, another postdoc in my lab, came to my office and said, let's stop this submission process. We want to see if we can use the platform to address the Ebola crisis, which was then at the peak of the crisis. And what the team did was remarkable. In 12 hours, Alex and Keith, along with others in our lab, modeled, designed, built, tested, and validated 24 different sensors for Ebola. 12 for the Zaire strain, 12 for the Sudan strain, each costing just a few pennies and could provide an output in about 90 minutes on a paper-based scheme. And so we were very excited opening up the possibility. By the time we got this out, the crisis had abated and did not have an impact on this, but we got a lot of interest from nation states around the world. And we're working on various pathogen implementations, bringing us to early 2016. And in early 2016, the leadership at MIT reached out to the community to ask who was working on Zika, as that outbreak was just beginning. We were not. We're not largely a virology lab, though we have repurposed multiple times, including for now SARS CoV-2. But rallied my team and said, let's go after this. And we did. And in now a matter of a few weeks, developed a highly sensitive platform using our RNA sensors shown here on the left, where we could, with high sensitivity and high specificity, discriminate Zika from dengue, yellow fever, chikungunya, other fever-based illnesses. And on the right, introduced what was then the first, still is, the first CRISPR diagnostic that used Cas9 to detect SNP differences between strains. In this case, the diagnostic had an impact on the outbreak. It was used in 7 different countries in 2016. We then launched a massive clinical trial in Brazil, Colombia, and Ecuador involving 3,000 patients. And just as the current pandemic took off, we completed that trial with great success, so showed that the platform could be used as an inexpensive diagnostic in the field, and those results are to be reported very soon. So what we have here is really a very flexible platform that enables one in a matter of a few days to program, design, build, and validate sensors for an emerging pathogen or newly emerging pathogen that is shelf-stable and can be distributed around the world, providing an output, a diagnostic test. Here we're showing 3 hours. We now have it down to about an hour. Of interest, I'm sure several on this call, I have many, many interactions with the US government, and even going back to 2016, had many discussions challenging them. So I should take a step back. I am regularly visited by people from the FBI, from the CIA, from the federal government, because we work on engineering biology, this broader topic of synthetic biology. And they're worried about bioterror. So will there be an engineered agent that will be made, that will be released or used for evil purposes? And no question there's a threat, but I emphasized again and again and again to the agents and to leaders that my worry has not been an engineered organism that's either intentionally designed and released or accidentally released, but what nature has in store for us. And like many of us on the call, was concerned about an epidemic, an outbreak, a pandemic, and said nature is so much cleverer than we are, and the next pandemic is coming. Back then I was saying, we don't know from where, we don't know when. Unfortunately, we are all now in this horrible situation. And frankly, an easy prediction to make is the next pandemic is coming. We don't know from where, we don't know when. It's likely going to be a zoonotic transmission. Something's going to jump from animals. We're increasingly pressuring these animal communities from crowded cities and folks living with livestock in increasing rates, and particularly developing world because of poverty. We need this, and I got a lot of pushback from the government that we didn't need to put it in a national and international capability, and I hope that our current horrible situation has changed that. Moving on is that we also showed, and of relevance to, I'm sure, a lot of interest on this call, that you could use these paper-based diagnostics to look at the gut microbiome, whether it was for nutritional health, whether it was for looking at infectious state, whether it was looking at inflammatory bowel disease, whether it was looking at infant health from a nutritional standpoint. And we've been using this in a collaboration with a group in Bangladesh for looking at an infant probiotic study. Here, what we showed is a piece we published just 2 years ago to show that you could couple synthetic biology with this paper-based platform in a very inexpensive, rapid manner to identify what species are available or present in a stool sample. So those of us on the call, on the webinar, who work in the microbiome, we typically will send things out for 16S sequencing. Very expensive and very time consuming. Here, we can do it for small numbers of dollars. Like less than $2 can give you an output in just a few hours versus taking a week and costing many hundreds, if not thousands of dollars. Further, we showed you could use this platform to get after host biomarkers. We could identify as the person in an inflammatory state by looking at human transcripts, and we could identify bacterial toxins such as that produced by C. difficile. Shifting gears, but in a similar spirit, when we published our work on the Zika effort that introduced the Cas9 CRISPR component, I was contacted by my colleague Feng Zhang, who is a faculty colleague with me here at MIT, and of CRISPR fame, one of the co-developers of CRISPR as a gene editing tool. And Feng's team was working on Cas13, which is captured here. Cas13 is different from Cas9 in a few ways. One, Cas9, which is the more commonly associated CRISPR protein, targets DNA first, and two, targets DNA in a highly specific manner, so that when it's brought to its targets by its guide, it will degrade with high specificity its target. Cas13 is different in that it targets RNA, not DNA, and when it finds its target, will degrade that target, but also have collateral activity and degrade any RNA in the region. So we launched a platform that we called SHERLOCK, after the British detective, that given a sequence from a pathogen or a host of interest, you could design guides to go after sequences in that target. When the guide brings Cas13 to that target, it'll activate Cas13, degrade the target, but also will then go after RNA linkers that are linking a quencher to a fluorophore, quenching the fluorophore signal that would then degrade the RNA linker, freeing the fluorophore and giving you an output. With Feng Zhang and his team and my team together, we were able to show you could get after a remarkably sensitive, rapid diagnostic that was much cheaper than PCR and could function in the attomolar range. You were now down below attomolar. We showed you could get after viral analyses, you could get after bacterial analyses, and you could embody this in paper-based lateral flow strips captured here, similar to pregnancy tests, but ones that could be distributed readily and easily in low resource settings. We were able to show also that you were not limited to pathogens. You could also do human genotyping. So we could give you an output in an hour to identify what SNPs does a human patient or an individual of interest carry. And so we showed this platform called SHERLOCK to get after pathogens and humans was able to be freeze-dried, leading to low-cost schemes. And we launched a company called SHERLOCK BIOSCIENCES that is developing this platform for many different applications. But of note, and with great pride, so I'm a co-founder and I serve on their board, we repurposed the company in the face of the pandemic to completely focus on COVID-19. And there are 3 major developments from SHERLOCK since that. 2 months ago, they announced that they used our SHERLOCK platform to produce the first FDA-approved CRISPR product, in this case, the diagnostic test for COVID-19. It uses this platform. It gives an output in under an hour, so it's considerably faster by almost an order of magnitude from a typical PCR test. It's a fraction of the cost of PCR, but actually has 100% sensitivity and 100% specificity. So it's as accurate and as specific as the PCR test. Second, and so we're very proud with that, is that the company has a major manufacturing deal now with IDT to make up to 1 million tests per week. The test cost of order about $25 to $30 in these limited ranges, but we think we can get cost of goods down to $2 and thus dramatically reduce the cost of the test as we scale up. 3, the company just 2 weeks ago announced a major partnership with Binx to take the COVID-19 test onto the Binx point-of-care device and produce a point-of-care device that can give an output in under an hour outside of a CLIA lab, so outside of a established, approved diagnostic lab, so it could be used in schools or at workplaces, and the hope is to have that ready by the fall. What my lab has done building on this is that, going back now about 2 years ago, we showed that this freeze drying of cell-free extracts and freeze drying of synthetic biology was not limited to paper, that it actually worked on any porous substrate, including plastic, including quartz, including clothing and textiles captured here. So our idea was, could we actually produce wearable synthetic biology diagnostics that could be used by healthcare workers, used by first responders, used by military personnel, used by global healthcare, individuals, hikers, athletes. And what we showed was that you could use more than 100 different fabrics and textiles to wick up cell-free extracts and wick up synthetic biology and CRISPR-based sensors that could then be freeze-dried and used in the environment. And we showed, captured here, that upper left is just a test to show you could have a constitutive synthetic biology circuit. On the right, it's showing that you can have, in this case, a rabo switch, so a synthetic biology circuit that is freeze-dried now into a wearable fashion that can detect small molecules. So might be metabolites produced by the patient or by a doctor or by an athlete or a military personnel. The lower left showing these RNA switches that our lab developed to detect pathogens. And here we're showing you can use it to detect HIV, that you're exposed to HIV. As a schema on the bottom right, we actually have developed a synthetic biology circuit that serves as a universal detector of nerve agents. And so if you've been exposed to a nerve toxin, whether as a innocent tourist walking around London or as a military personnel in a hotspot, you could get an easy readout to know that you need to seek medical treatment as soon as possible. We initially, and have been pushing to incorporate this technology to create the lab coat of the future for doctors, nurses, and other healthcare workers to indicate if they've been exposed on their rounds to the hospital to different pathogens to then help better contain and identify an outbreak in a hospital. We're moving towards making smart Johnnies for patients as well. So, patient in a hospital is often wired up, making measurements on various physiological signals. Here we're looking to see, can we extend that to also report on pressure sores or wounds and identify, are those infected, and if so, is there a resistant pathogen to give readout? Again, it's kind of a common theme here. We were advancing various embodiments when the pandemic hit and got excited to see, could we use this technology to create a wearable diagnostic for SARS CoV-2 and specifically to use it in the context of a face mask. So what we've been advancing, and we're very near to finishing our academic prototype, is the idea that you can have freeze-dried synthetic biology elements into an insert that can be added to any face mask. They would then be worn by an individual for an hour or two. And the normal act of breathing, talking, coughing, and sneezing will release viral particles if one's infected. And the idea would be, at a high level, that this synthetic biology insert would collect, process the viral particles and give a fluorescent readout if the person's infected, one that couldn't be seen by eye but could be detected by handheld fluorimeter. What we've done, that's shown schematically, is actually capture and design a system that is a multi-layered system that can capture the sample, process it, and give a readout. We've already now built this and tested this, so this is captured here. It's actually a foldable, paper-based lateral flow microfluidic system that can capture viral particles, lyse them, amplify the RNA, and then apply it to our synthetic biology constructs. And we're getting beautiful output at levels, detection limits that are appropriate for this outbreak. We've designed test equipment systems in our lab at MIT and Harvard that enable us to ensure that the insert does not prevent the person from breathing, as well as various test equipments to see how it functions in the face of aerosolized viral particles. We've now actually implemented a number of CRISPR sensors that are giving us beautiful sensitivity in, again, this wearable scheme. We are taking this exact technology as well, or in the final stages of developing an at-home saliva-based test using the same scheme. This would be an idea that you'd wake up, you could just stick the device in your mouth, and it would give you a readout at home, similar to a pregnancy test, but in this case, you wouldn't be peeing on the stick. You would be sticking in your mouth, which would then process your saliva in appropriate time. And in each case, we're maybe just a week or two away from an academic demo and plan to work with SHERLOCK Biosciences to translate this, and of interest maybe to many. On the call, I was actually on with many members of the Indian government and health department this weekend talking about how these technologies could be translated, potentially used in India and around the world in the face of the pandemic, and it's something we are aggressively addressing. Briefly, we also have shown that this freeze-dried scheme is not limited to paper, clothing, or plastic. That you can actually freeze-dry the pellets, freeze-dry the cell-free extracts as pellets, freeze-dry the synthetic biology components as pellets with no substrate. And the idea is you can then have these in a small container that you could use and distribute around the world by simply then adding water to produce biomolecules of interest in the field. And so these would consist, for example, of antimicrobial peptides or vaccines or antibodies or nanobodies. And it turns out that this works beautifully. That here we show you can freeze dry the machinery to make antimicrobial peptides that you then just simply add to water in the field, and they can then be applied to a pathogen. Stunningly, we showed you could use this with vaccine antigens. So we've not demoed it on the COVID-19 just yet, in part also we don't have an approved vaccine, and I'm going to turn to that in a couple of minutes. But that you could use this as a means to now, in this case, produce diphtheria toxin vaccine antigens that were biologically active. We could deliver them, we only did in animals, but in the field setting, that actually could produce an immune response that was appropriate. Again, now removing the cold chain, so you can have this in your pocket as a healthcare worker, open it up, process it with water, with just a small number of minutes, and then deliver it as needed to the patient. We also showed that you could use this in a multi-step fashion to produce different small molecules of interest. And that is that these could be made so that, in this case, we looked at the violation pathway. That's a multi-step pathway to produce different small molecules that involves 5 different enzymes. And we were able to show that you could freeze-dry the cell-free extracts and freeze-dry the 5 different enzymes. And then in a mix and mash fashion, add these to water, a small amount, a large amount, and produce each of the 5 small molecule outputs of the system. And so we're very excited about this is for the current pandemic, for future outbreaks, epidemics, and pandemics, for just even general use in the field, that here's this inexpensive means to produce molecules that are needed on a case-by-case basis. I want to turn briefly to vaccines with 3 slides. So of maybe interest to many on the talk, my lab actually helped co-develop the founding technology from Moderna. So Moderna's got a lot of interest in this country for their vaccine efforts. I'm not sure how much is happening or what you know in India, but they've been racing fast using their technology. So the technology that we developed with Derek Rossi and George Daly was this idea that you could use synthetic biology to create synthetic mRNAs that would be stabilized so they wouldn't be degraded too quickly, and that they wouldn't produce an immune response. And we showed that you could then deliver these to living cells in the cytoplasm that could then make a protein or multiple proteins. And we initially did this for cellular reprogramming and cellular differentiation. In this paper that was highly cited in Cell Stem Cell presented this. And of note, if you go look at the paper, and encourage you to look at it, it's a very nice paper. In the discussion, we talk about how this platform could also be used to produce RNA or DNA-based vaccines, as well as various therapeutics. And this is what Moderna was founded on, and they're now using this technology, slightly modified, to deliver synthetic mRNAs to human cells that are encoding the spike protein from SARS CoV-2. And they're programmed then to produce the protein and trigger an immune response. And early phase 1 trials by Moderna indicates that it's safe and appears to be producing antibody levels. And the hope is that this and others will get us to a vaccine sooner than later. And Moderna's plan is to launch a phase 3 trial this month, and we're hopeful they will. As mentioned at the outset, we are also working on a different synthetic biology approach to the vaccine. And of interest maybe to many on the call and the interest of the longstanding use of BCG in India, we're actually repurposing the BCG vaccine. So the BCG vaccine has been used for almost 100 years. It has one of the best safety profiles of any vaccine in the world, and it's easily manufacturable. It's a live attenuated vaccine, so the BCG vaccine is based on Mycobacterium bovis. This is a bacterium that causes essentially TB in cattle. It doesn't cause TB in humans. It's a cousin of Mycobacterium tuberculosis, which is a pathogen that causes TB in humans. It has been used as a TB vaccine, as I said, for almost a century. It's still delivered, I think, to about 100 million people each year. Of relevance to the current pandemic, there are a number of trials underway around the world to see, does this vaccine, without any modification, provide some protection against SARS CoV-2? And there is indication that it does. And what's intriguing is that the notion is that the T-cell response that it triggers might provide additional benefit. Where we are going is with Andres Cubelos, one of my star postdocs, and his brother Juan Cubelos, who's on faculty at Cornell Med, is we're taking a synthetic biology approach where we have engineered M. bovis to express on their surface various SARS-CoV-2 antigens, including the spike protein. And we've now shown you can do this with very high efficacy. And we have animal studies underway, and I hope to have results to report in a week or two. And we're very excited that with this, we may get kind of a two-pronged benefit. One is that we would trigger an immune response specific to SARS-CoV-2, given the SARS-CoV-2 antigens expressed on the surface of M. bovis, and we would get the secondary immune response, the innate immune response from the T-cell stimulation. And so again, more to report soon. Moving ahead, and just to kind of finish up, so maybe we can leave some time for discussion if that's allowed, is that we've also now been turning to deep learning in two different ways. And I'll maybe just give high-level discussion. One is this idea of design, building, and testing engineering biology is very exciting, but invariably the circuits that we make don't function in the way that we want. And so you need to turn to post hoc tweaking, where you tweak the system again and again to see if it behaves in the way you like. And that can take many weeks, many months, and in some cases, years. We're now turning to deep learning. And I'll probably end on this slide. It's going to be animated to explain what we're doing on this, and then to open up discussion also on therapeutics, and then we'll turn to questions. The idea here was, could you use deep neural nets that could be trained on data that you could synthesize synthetic biology components, sequence them, functionally characterize them, train the network to learn the rules, the design rules, going from sequence to function and composition to function that are present in nature, and then use that to infer the design rules, make novel components, and make novel gene circuits. We've now done this. We've done this specifically for the RNA sensors that I shared. We generated 100,000 circuits, sensors that we synthesized, characterized, sequence and characterized, and trained the model to learn the design rules. We then used it to actually design better sensors for SARS-CoV-2. And this work will be coming out soon in Nature Communications. We then went further, and I think there's much to be done in synthetic biology, but turning to therapeutics. We also wanted to see, could you train a deep learning model, kind of captured here, to address antibiotics? And briefly, antibiotics, as I mentioned earlier, is a big focus for my lab. And it's actually relevant to the current pandemic as follows: that bacterial co-infections are hugely problematic for COVID-19 patients. 1 out of 7 COVID-19 patients who are hospitalized have a bacterial co-infection. 50% who die have a bacterial co-infection. And so it's incredibly important, can we come up with effective antibiotics? And what we did in work that was published in Cell in February, we showed you could train a deep neural net on a small number of molecules to associate and learn features of antibiotics that could then be applied to large in silico libraries. And we've identified now a remarkably new set of potent antibiotics that can go after multi-drug resistant, pan-resistant bacterial pathogens. And we now are going after narrow spectrum and using these deep neural nets to design molecules de novo. The platform is not specific or limited to antibiotics, and in the midst of this SARS CoV-2 pandemic, we've now also repurposed this to train deep neural nets to identify antivirals. And challenge is getting after the right training data, but we now have sufficient training data to get after early predictions. And for those of you interested in deep learning, we now are moving this towards getting after combination therapies. I think our way out on the therapeutic will not be single agent treatments, but will be on identifying combinations. And the challenge is that in combinations, the geometric explosion of possibilities makes it empirically impossible to consider all such possibilities. But deep learning, where you can train the model in a computer and then use the computer to process through millions, billions of possibilities in a matter of days, becomes now eminently plausible and possible, and that's where we're now moving. And so with that, why don't I now open it up, and hopefully we have some time for discussion.
Thanks, Jim, for your nice presentation. And now the session is open for the questionnaire queries. I'm asking all the participants, if you have any query, you can ask it directly. I'm in the chat box. It is visible. One question is visible, that how to work sonication in the freeze-dried cell free synthetic biology. Can this process also work for HIV virus?
Yes. So in terms of detection, absolutely. We are working with Bruce Walker at the Reagan Institute to develop a very inexpensive diagnostic for HIV for various applications in HIV. One of the challenges is to really get after very low detection limits. And there it's how do you do it inexpensively and quickly on the amplification. And we've got a number of efforts now around rolling application. And soon we'll be reporting an effort of a freeze-dried HIV diagnostic.
Okay. Can we use the synthetic cells in case of drug designing?
So interesting on a few counts. One can absolutely use the cells as drug factories, and in fact, portable or deliverable drug factories. And that's what we were showing earlier with the pathogen work, and is really the focus of SynLogic. We are using our cell-free extracts around drug design, both also production for remote efforts, but also for design. Getting after your question is interesting, and I'll share very new work from ours. From the idea of design, we now have an effort that's just about to be finalized for Nature Communications to show that you can use living cells to better evolve or quickly evolve antibiotics against a resistant strain. So there it's technically not designed in the usual engineering sense, but it's using, in this case, the power of biology, specifically the ability of these organisms to evolve, to rapidly get after the design, I'll put that in air quotes, the evolved design of an improved molecule, a therapeutic molecule.
Okay, thank you. Can we use synthetic cells to produce insulin at an industrial level?
Yes, we can. And in fact, that's really at the heart of biotechnology going back decades ago, where in the early efforts, so-called genetic engineering from the 70s through to about 1999 really had very little engineering. It was effectively transposing, introducing a gene from one organism into another, engineering that gene to make a molecule. And insulin is one of the more famous biological molecules made by the biotech industry. But getting maybe now at the synthetic biology of it, there has always been a considerable interest on could you engineer a human cell or a synthetic cell, even a bacterial cell that could make insulin to create an artificial pancreas or a cell-based insert that could be used. And there's been some very nice work by Martin Fussenhager from ETH in Switzerland doing just that. I've been intrigued and have had aspirations in those directions, but my group has not made it as far as Martin has for that specific application.
So the next question is, can this technology be used in the cancer treatment and screening? Yeah, I think so.
So that, again, very interesting question. Yes, and in fact, SYNLOGIC, the company I mentioned, has a clinical trial for cancer treatment where they are engineering bacteria that could be injected into a solid tumor, and those bacteria basically stimulate or take advantage of the STING pathway to disrupt and kill the solid tumor. There are a number of efforts underway using engineered bacteria, led primarily by Jeff Hasty, who was one of my early trainees in synthetic biology, to engineer bacteria to serve as a diagnostic or a screening tool for cancer that you could pass through the body to identify that you have a tumor, liquid or solid, and are in need of treatment.
Someone is asking that are all these synthetic technologies nature friendly? It's the great question, I think.
Are they what? I didn't catch the, are they what? Are they?
Are all these synthetic technologies nature friendly?
Nature friendly. You know, it's a very good question. I think the majority of them
exactly
are nature friendly. The majority are really just kind of swapping things around, or they're largely harmless. And nature remains this, very large, amazing force that will push back on any perturbation. Having said that, there are worries of untoward uses of this. Could you make something that would be profit motive, that would force somebody to buy your product? Would it be something that could cause harm to humans or cause harm to their crops? The answer to each of those, unfortunately, yes. There's concern that, could you have accidental release? Or could you, for example, in the work that we do, have the intent to make something that would help the patient, but maybe it has an unexpected side effect? So to address that, we have led many efforts to create safety switches and biocontainment systems that could be activated if there is an accidental release. So that could be activated if you have an untoward side effect, or it could be activated just as it leaves a patient's body that would be intended because it may be passed through the GI tract. And each of those works very well, thankfully. But there is still a lot of work to be done and open discussion needed to consider as these new technologies be developed. And I'll end this answer with a comment in a second, to make sure we're doing this in smart, safe, and acceptable manners. And my comment is that I think synthetic biology will become one of the defining technologies of this century. So for the young people on the call, it's a tremendous space to get into. I think India has a very big role to play, both on the computational and the experimental end, given the extreme high-level technical talent that's being trained in India. So when I look on this century, I think in the coming decades, we're going to look back to see that synthetic biology and artificial intelligence are the two dominant areas. And in each case, we need to engage in appropriate ethical and social impact discussions from the very beginning to ensure that it's nature-friendly along the way and human-friendly.
The next question is, I'm coming to the next part of the question, is that one of my friend and academician, Dum Dum Motijheel, is asking that for the wearable synthetic biology, how sensitive is it in terms of the viral load?
Yeah,
in terms of the viral load.
Yeah. We have been able to detect in very low femtomolar. So it's actually at a highly relevant level for exposure. And on par with actually many CDC guidelines. So it's remarkable actually where we can get to. And part of what we do is that we use a room temperature nucleic acid amplification step. So you get a readout. It's not an instantaneous report. It's still after several minutes, but we use this isothermal room temperature amp to get us into the low femtomolar.
Another question is that, can we use antisense mRNA technology to fight against a certain pandemic?
Very interesting. So I didn't share, but we actually have a therapeutic effort underway from a synthetic biology standpoint where we have targeted different regions of SARS CoV-2 genome, specifically in their IRES region, to related IRES region, to modify and disrupt their replication. For reinfections, that is within the same host, we've shown you can get upwards of 90 to 95% suppression. Sounds pretty good, but it's probably not good enough. Now, I think it would probably serve to reduce the symptoms. But the idea is that these would be very small. So we actually are doing with very short oligo, shorter than antisense. And so easy to deliver, easy to aerosolize. Intriguingly, we've gotten pushback from the biotech and pharma industry in that their interest is primarily getting after small molecules versus the nucleic acids, but nonetheless, there's a lot of potential there.
Next, the Shovik Chakravarti is asking that, how can we find the complete evolutionary biology of the coronavirus using the synthetic cells?
So how can we get after, you were saying, using the synthetic cells to get after which question?
Exactly, the question is that how can we find the complete evolutionary biology? Probably he's asking for the origin, searching the evolutionary biology from where it is coming. I think he's asking about the origin, trying to find out the origin through the synthetic biology.
So I guess I'll maybe look at it, I'll answer it in a couple ways. You know, I do think synthetic biology has the potential to get after biodiversity mining, and that is finding the origins of different components that are being used. Second is on kind of the evolution. There is a very active space in the field of getting after origins of life studies. So can we understand how cells evolved, how components evolved, and folks like Jack Shostak and Jerry Joyce are leading various efforts to use synthetic biology frameworks to really get after hypothetical frameworks that the system evolved in this way in order to address different functions. And so it's a very exciting space. We've not engaged in that given our biomedical focus.
One of my friend is asking, Orijit Chaudhary is asking that, can the synthetic biology, can the synthetic technology be used for a common platform for various RNA virus vaccine?
Yeah, I think so. You know, I like your thinking. And I think that's at the platform or the basis for Moderna. I think they're hoping that theirs will function well. But related, you know, shifting from an RNA vaccine, I also think that this BCG chassis of sorts will also potentially serve as a generalizable scheme for getting after other viral vaccines. And that you can imagine that if you identify the appropriate antigens, which don't need to be singular, that you could rapidly engineer BCG, which grows slowly, albeit, to express those antigens on their surface. You could also have that as a problem. So I think synthetic biology is offering two generic, reprogrammable schemes that should be applicable to a broad range of viral pathogens.
One is asking about, one of our students is asking about the side effects of this approach of the synthetic biology.
So, you know, I presented many different schemes. The synthetic biotics, it's interesting, we've seen very few, if any, side effects throughout our efforts. Synlogic, you know, the one potential side effect is that when you're actually taking a probiotic, if you're taking a large amount, you might have a little bit of nausea because you're passing it through the stomach. But no untoward effects then as it passes through the GI tract. I suspect in each case it's going to be important, as of course, to do a clear safety profile, including looking at side effects that may be untoward experiences by any given patient.
Jim, now there is a very interesting question. Everyone is asking that do you think SARS-CoV-2 genome is a result of synthetic biology? It's a very controversial question.
No, I actually, well, I don't, my honest answer is I don't know.
Exactly.
You know, I think we all really want to get after where it was so we can better understand, you know, how do you prevent this? You know, the genomic analyses, you know, indicate in the spike protein, there is a very interesting sequence that makes it very easy to enter human cells. My worry with no data is that were there gain of function experiments being done where either the virus was being evolved to see if it could pick up this insert, or was the virus engineered to have the insert and then explored in a lab around the world. But, you know, let's leave it broadly at that. I don't know.
I would like to add something in that
yeah, sure.
Probably it has a natural origin, because the furin structures and those structures, it is already published in a paper, Nature paper. I think that is quite convincing to say that, prove that it has a natural origin. It is not synthetic. I think so. I think so.
Well, so I want to, where I say I don't know, and I'll just push back a little on that, is that, yes, so the sequence is natural, occurs in nature. It does not, is not commonly in coronavirus, on this, it's found in other viruses. But of note, so synthetic biology is not simply a matter of synthesizing, pulling a new sequence. It's often taking what's natural and putting it in. That's one. Two is, there are reports of colleagues of mine at UNC and colleagues in Wuhan that were interested in looking at this sequence, specifically in coronaviruses. And so it's not impossible that somebody considered inserting into virus to explore it. It's not impossible somebody explored, could you evolve it to pick it up? Again, the honest answer is we don't know. It's likely just natural, and that so then the next question is, was it being studied in a lab, extracted from a bat, or was it jumped through one of the markets? Again, we don't know. And unless somebody comes forward and says they were studying it, and here's my record, the easiest is you default to what people are reporting, which is likely that this evolved naturally, picked it up, and jumped from bat or another organism into human, and now we're dealing with it. Likely is always the better thing to go with, but the real complete intellectual answer is we don't know.
Okay. So we're coming to the next part of the question, is that can CRISPR technology be used anyhow in the COVID-19 treatment?
Yes, I think it can. And as I shared, we're exploring a number of different possibilities there. And, at a high level, I should say, I think we will either have some level of herd immunity, or the virus having its run through different communities, or a vaccine before we arrive at an effective therapeutic.
Okay. Can we use synthetic biology to predict the decrease or increase in the virulence of the coronavirus? If so, how it is possible?
Yeah, I don't know. I don't know that you could do it to predict it. You certainly could envision using synthetic biology to explore in a very systematic way how different modifications to a genome could influence virulence or transmission. The challenge with that really gets after the earlier question on, was this from a lab or was it being studied, is that for the young people, there was a very famous case of just a few years ago where people were looking at a respiratory virus, doing gain-of-function evolved experiments around transmissibility, and identified some number of mutation sequences that would dramatically increase the transmissibility and potentially impact virulence of a given virus. And there was massive debate around the international community, should those sequences be published, because of the worry they could be used by people with evil intent. My worry for that question is the trade-offs on, as you learn more, could it be used for evil intent, and or do we have enough safety checks in place that as we study it, you don't have an accidental release or an accidental infection of a poor person just trying to do their job of studying it.
There is another question. Paulomi Mukhoda is asking that, is detector being used in SARS CoV-2 detection like SHERLOCK?
So SHERLOCK, I was curious on that last comment.
Yeah, is detector being used in SARS CoV-2 detection like SHERLOCK?
Oh, yes, it is. So,
yes.
So detector is a very closely related platform developed by Jennifer Wadman's group at Berkeley. That primarily gets after DNA than RNA, though ours also goes after DNA. And Jennifer launched a company called Mammoth Biosciences that's also getting after a broad range of diagnostics, including a SARS CoV-2 effort. And they have a partnership with GSK to see if they can use their CRISPR platform to develop an at-home test.
Now, another question is that, can synthetic biology be put to use for treatment apart from the testing purpose? Yeah, I think so. You are absolutely.
Yes, absolutely. So we actually have trials underway where it actually is as a clinical, and the hope would be that it would serve as a new class of medicines and not simply used to get after new basic insights in a lab, but it would be a new set of medicines.
Now, at the end, Collins, Jim, I just want to ask you one question. It's from my part, and
Yeah.
that is, I think you were interested to work with the BCG vaccine, the repurposing of the vaccine, and if, as long as my information is concerned, the BCG vaccine cannot work on the adult persons. So, I just want to know in which part you were using the synthetic biology for the repurposing of the BCG vaccine, and how can it cover the adults rather than the infants in case of COVID-19?
Yeah, no, it's a good question. And we are hopeful that for our application, it will be applicable to adults, including older adults. So we would not be limiting it to infants in the way that it's used for TB. And so here we'd be getting after all ages with the idea that you still in adults trigger a T-cell response, as well as now we would be expressing SARS CoV-2 antigen specific. So no, ours is to look across entire age ranges.
Another question I would like to ask you is that, can your proposed vaccine or can it cover globally? That means different strains are there. These strains, if the vaccine is completely oriented to a particular strain or a particular protein, it is mutating itself very first. So can it cover the single dose of that vaccine can be useful to combat the COVID-19, or the booster doses are required?
Yes, I mean, at a high level, mutations can be a feature that we can use in directed evolution, but it's also a feature of biological systems that really poses challenges for synthetic biology. One, to see that our circuits function in the way they should when we need them to function. And then second, to your point, can we use synthetic biology to address the, say, mutations effects? Now, luckily, the current SARS CoV-2 appears to have a very low or slow mutation rate. That's not been well addressed yet. So yes, synthetic biology, because we can rapidly reprogram schemes, you can envision reprogramming the vaccine to adjust for a mutated spike protein. You still have to have the regulatory challenges. Is it still as safe? Is it still as effective as the earlier version? I remain intrigued and would challenge young people on the call to think about, could you come up with synthetic biology platforms that would evolve your therapeutic in the face of the evolving pathogen? So could you have them going hand in hand, so to speak, and making changes? And I've been intrigued on that, and we've yet to have anything really come out of it.
Is there any other question from the part of the participants I would like to ask? Another question our student is asking that, can synthetic biology be used to prevent a re-wave of the COVID pandemic in future?
I think that synthetic biology can help prevent and or ameliorate the next wave, and also put us in a good position for, as I alluded, the next pandemic, really on all of the things I shared. I think the field is offering inexpensive, easy to program, rapid diagnostics. It's offering the ability to get after clever diagnostics, and also, as we alluded, both at the RNA level and these live attenuated schemes, rapid programmable vaccines to be developed. So I think this idea of engineering biology dramatically expands our capability to address this pandemic in all its forms, and also the next outbreak, epidemic, and pandemic.
Yeah. Another question is that I think it is already addressed. Can this technology find a way for different strains of COVID-19 cause their mutation rate is too high? I think you have covered this question. We have discussed.
I think it's possible to, again, rejigger both the diagnostic, a potential therapeutic, and a vaccine in the face of mutations. You still would have then the downstream approval processes through, for example, the FDA or its equivalent.
Yeah. So is there any other question from the part of the participants? Okay, there is another interesting question. Can nanotech be merged with this synthetic technology and AI in curing disease like cancer or pandemic situation, or like curing HIVs?
Yes and yes. On the
Yes.
latter, I alluded that I think a dramatic emerging space is AI, and also the integration of synthetic biology and AI for diagnostics, for therapeutics, broadly for the field itself. I think on the front end, you can very much view synthetic biology as a biological version of nanotechnology, given the scale we work at. But it's actually an underexplored space as to how you can couple synthetic biology to inorganic nanotech. And again, to the young people on this webinar, I encourage you to think about how one could use that. It's a space to be explored with, I think, tremendous intellectual bounty to be harvested.
Okay, so can we create biological robot by using synthetic biology?
I think we can, and a couple counts. So I encourage you to look at some work by George Church that has created so-called nanobots or biological robots using synthetic biology. We are looking at creating synthetic sentinels using engineered liposomes to encapsulate cell-free systems and synthetic circuits that could pass through environments, including the body, making measurements, making decisions, then acting on the environment. And you can view that in some ways as a robot. Now, getting after the mechanical schemes, I do think there's interesting Brownian ratchets and DNA elements that could possibly be used.
Okay. Can it help cure disease like diabetes and cancer? We think we have covered the part of cancer, so can it be used for the diabetes, this synthetic biology approach?
I think it can, and it's been under-addressed. So again, I encourage young people to think about how one could utilize it in different embodiments. But I think it can very nicely.
Another thing, one of our students is asking that what is the scope of synthetic biology in pregnancy? Can it ease several complications that arise during the delivery of the baby?
Yeah, it's not. I've not seen real application on the pregnancy side yet. You could envision how it could be very useful as a monitor and as diagnostics, so monitor to report on the mother's health in the process, but also on a diagnostic throughout the process, on maybe reporting on the infant throughout the birth process. So, Bibiswan, I need to jump off to go on to a COVID task force. So if we could maybe end with maybe a final question would be useful.
Yeah. Does synthetic biology necessarily mean that life can artificially be created?
No, it doesn't. It really, right now, the field, it's not so much about creating artificial life, which we've not done, and very few groups, I think, are anywhere even close to it. It's more so that it means that living cells, living organisms can be re-engineered, repurposed, redesigned, modified for various purposes.
Okay, thanks, Jim. Thanks for this nice presentation and deliveries. This is such a learning process. And these new techniques, you have introduced these new techniques, and you are an expert in this field. So we have the heartfelt gratitude for you, and we are really honored that you have given us this time for the discussion, and we are enriched. So thank you very much, Jim.
Well, thank you, Bibiswan. Thank you for this marvelous platform and the invitation. I want to thank everybody for participating, and thanks for the great questions.
Thank you very much.
Okay, good night, everyone.
Good night. Good night. Yeah. So we are almost at the end of this session, and not only the session, we are almost at the end of this 2 days webinar series. So we will, yes, some people are asking for in the chat box that for the feedback form. We will almost at the end of the we are almost at the end of this webinar series. So but you have to be patient for a few minutes more. We have some official announcements, and we have some conventions to end of the session. And at the end of this session, we will provide you the feedback link in the chat box. Please be patient for a few minutes more. Now, one of our very senior and respected teacher, Professor H. Shah, asked me to say something to the participants, and I am requesting Professor Shah to say something to the participants. Professor Shah, over to you. Professor Hadun Shah, are you there? Professor Hadun Shah, are you there? Okay, if he joins again, then we'll heard something from him. And now I am requesting our IQAC convener, S.K. Roy, Dr. S.K. Roy, to say something, says a few words about this international webinar. And then I will ask our principal sir to say something. And then I will address the formal vote of thanks. And then it will be wrapped up. So now I will ask Dr. S.K. Roy, the convener of the IQAC in our college. Are you there?
Yes.
Yes. So please address the participants.
Thanks to all the participants and the organizer, and thanks at all the research persons. It is very interesting for our college, in particular this situation of COVID-19 protocol. Our college, in particular the microbiology department, has done a great job to utilize these times for the teachers, students, and actually all the stakeholders. I think it will be very helpful to deal with these students, in particular all the teachers and students. I thanks again to all the participants, and I think it will be very utilization to all of us. Thanks again, not more. Thanks. Over to thanks. Bivasan, please, over to Bivasan.
Now, I would like to invite our head of the department, Dr. Bibosha Mondal, to deliver the vote of thanks.
Well, Dr. Mondal, I do not hear from you.
Bivasan, a principal is here. Please ask to vote of thanks for him.
I do not hear from Bivashan. I think there is some technical problem is there.
I think there is some technical problem. So, Principal Sir, it's over to you.
Okay, sir.
I think Bivashan has completed his vote of thanks. So,
okay.
Not yet.
I think now I am audible. It was actually not.
Yeah. Now you are audible.
Okay. First, principal sir say something, then I will go for the vote of thanks.
No. I think you started your vote of thanks.
Okay. Sorry, sir. It was, I think the microphone was off for a few minutes. Okay. Sorry. So Respected eminent speakers, respected principal sir, Dr. Pradipta Gupta Roy, respected convener, IQAC, Dr. S.K. Roy, respected convener, seminar committee, Dum Dum Motijheel College, Dr. H. Shaha, my dear colleagues of different educational institutes and my own institute, researchers, doctors, and many my beloved students. I consider it a great privilege to propose a vote of thanks to all the dignitaries who have witnessed it as a memorable and successful event. I express my heartfelt gratitude and sincere thanks to all the eminent speakers who are the national and international experts in this field of science. We really feel honored by their gracious presence in this webinar. I am thankful to all the participants for attending this webinar. More than thousands educative minds from different disciplines registered for this event. With available cloud platform, 250 participants attended each session of this international webinar. We express our sorrow for such technical inconvenience, but hope the attendees learned many things about this pandemic disease. Professor Bashop Chaudhary, Honorable Vice Chancellor for our university, sir, our gratitude is due to you for the excellent inaugural address with which you inaugurated the webinar. I take this opportunity to express our sincere thanks to our principal, Dr. Pradipta Gupta Roy, the dynamic and flexible person in all academic matters, for giving this occasion. We are extremely grateful to you, sir. I express my deep gratitude to our respected IQAC convener, Dr. S.K. Roy, and former convener, Dr. H. Shaha, for guidance to us in all activities. Ensure that we always do the right thing in the right way. I acknowledge the unwavering support received from the fellow colleagues of my own department and other departments of our institution. In particular, I would like to acknowledge Srimati Priyanka Mukhopadhyay, Dr. Sahawaj Anwar, Srimati Jhulan Basu, and Dr. Kaushik Rai for their constant support. Finally, the wonderful students, not only of our department, but also from other departments, and not only of our institutions, but also from different other institutions. We are thanking you for so much interest. So thank you so much to all the participants. Thank you very much. I think we are just at the last of this particular 2 days international webinar. Now I am going to share the link for the feedback form for this session 4. I think it is shared. I think it is shared. The link of the feedback form is shared in the chat box. Yes, some people are asking that can we leave? Yes, ma'am or sir, we have concluded this session. If I think the link is working, if there is any problem, some people are saying that it is working. Some people are saying that it is not working. For them who are saying that it is not working, please check your connections, please.
Yes, it is working absolutely fine, Bibhushan.
Yeah. Thank you. Thank you very much. And I just officially announce another thing, very important. Some people are asking me for that certificate. Yes, all the participants who have participated in this webinar, we will send you the certificates through your email, but we need more time. We need at least 1 week for this certificate. Please be patient. We will send the certificates in your corresponding emails. Thank you. আরে কি করে প্রেস বন্ধ হয়ে গেছিল ওখান থেকেই এটা। হ্যাঁ হ্যাঁ বলে আমি কিছুটা বলে ফেলেছিলাম আমি সামনে। হ্যাঁ তারপর স্যার বললেন মানে প্রিয়াঙ্কা সঙ্গে সঙ্গে এ করলো এখান থেকে, তারপর স্যার বললেন হ্যাঁ সবাই লিভ করছে।
Sir, when will we get the certificate?
Ma'am, probably within a week we can be able to send you the certificates through your corresponding registered emails.
Okay, thank you, sir. I have participated today, and I have also given my mail ID. So please kindly send the certificate.
There is no problem,
Okay,
ma'am.
thank
We will provide you the certificates. There is no.
Okay.
It is not an issue, ma'am. Okay.
Okay, thank you, sir. It was an excellent session, and you have organized it very well. Thank you so much.
Thank you, ma'am. Thank you, madam. It's our pleasure, ma'am.
Okay, sir, I can leave now?
Yeah, you can. You can.
Okay, thank you, sir. Good night.
Good night. হ্যালো হ্যালো
হ্যাঁ বিবর্ষণ বলো।
হ্যাঁ আমাকে প্রিয়াঙ্কা কি পড়ে গেছে
না ওর ফোনটা হ্যাং হয়ে গেছে বললো। হ্যাং হয়ে গেছে বললো। আমিও হয়ে গেছিলাম, আবার দেখি হঠাৎ চলছে। আমিও হ্যাঁ আমারও ওরকম হয়েছিল।
ঠিক আছে, আমরা আর কিছুক্ষণ বেরিয়ে যাই। যদি কারো
কিছু কি মিস করলাম নাকি?
না
কিছু বললে?
না
ওকে ঠিক আছে।
আর কিছুক্ষণ থাকি, যদি কোনো পার্টিসিপেন্টস এর এন্ড থেকে যদি কোনো
প্রশ্ন থাকে ঠিক আছে। ঠিক আছে।
ওয়েলকাম আনোয়ার স্যার, ওয়েলকাম।
সবারই মনে এক হয়েছে, সবার এক হয়েছে।
প্রত্যেকে ভালো হয়েছে স্যার।
আনোয়ার কিছু বললে?
স্যার বলছি অনেক ভালো হলো, সত্যিকে ভালো হলো।
ওকে এটা তো আমাদের সবার ইফোর্ট।
আপনার জন্য হয়েছে স্যার।
তোমার এফোর্ট সব থেকে মানে কি বলবো 100% এফোর্ট তোমার। আমরা হচ্ছি সহযোগী। হ্যাঁ, তো তোমারই সাকসেস। আর তুমি অ্যাচিভ করেছো, তুমি যে পরিশ্রম করেছো সেটা তুমি অ্যাচিভ করেছো, মানে একদম এক্সিলেন্ট একদম।
দেখো এগুলো একজন একটু ড্রাইভ করে ঠিক আছে,
আমরা আর কি আমরা কিন্তু পিছনেই বসেছিলাম।
আমাদের এই তো প্রিয়াঙ্কা চৌধুরী চলে এসছে,
আমাদের ম্যাডাম চলে এসেছে।
ম্যাডাম মিউট করে আছেন। ম্যাডাম আনমিউট করে কিছু বলবেন?
চলে গেছে।
আমি তো বলেই যাচ্ছি। না চলে যায়নি, আমার ফোন হ্যাং করে গেছিল।
হ্যাঁ বুঝতে পেরেছি।
বিভূষণ খুব ভালো হয়েছে।
ও থ্যাংক ইউ।
পরিশ্রম করেছো বলেই এত সুন্দর হয়েছে।
আচ্ছা নন্দিনী
একদম
স্যার ঢুকতে পারলেন না তো আজকে দেখলাম।
স্যার ঢুকতে পারেননি। হ্যাঁ স্যার
লাস্টে স্যারকে আমি হোয়াটসঅ্যাপ করলাম, কিন্তু স্যার বললেন আমি ঢুকতে পারছি না।
দিলা তো? হ্যাঁ
না কিন্তু